Well, thank you all for coming, and Happy New Year, and welcome to the January 2025 meeting of Science, Scripture, and Reason. And we are really privileged, so I think we're going to have to borrow Lester's word. We're going to have an amazing presentation tonight. And because it's a really full presentation, I'm going to abbreviate the bio. You've all gotten that on a message and an invitation. So just in interest of time, then let me do a quick summary. Lester is a semi-retired engineer, and he has a long-standing history in math and science, and therefore a passion to figure out how things work. In addition to his work, he's been very active in Reasons to Believe, both as an apologist and chapter leader, and has also spoken to a number of churches and schools about Christianity, and has amazingly also has a monthly meeting for skeptics about Christianity. So one of his areas of interest is DNA, and we're going to be having the privilege tonight of hearing what he has to say about DNA, about the structure, the enormous amount of information in DNA, as well as the intricate and elegant biomachinery that's needed for both the replication, repair, and the transformation of information into proteins, which are therefore life-giving. So, and all this showing the signature of God. So, Lester, I'm going to give you the controls, and thank you very much.
Okay, thank you, Randy. Welcome, ladies and gentlemen, from Canada. It's about 0 degrees Celsius, which is 32 Fahrenheit for you. This is a topic that has really deepened my appreciation for our Creator in terms of the amazing features of DNA. So when I compare our Christian worldview versus the atheist worldview to explain the origin of life, the features of DNA point to the God of the Bible with the overwhelming weight of evidence, in my opinion. Just to let you know that I'm not a biologist, but I have come across the amazing features of DNA that some of you may not know. One advantage I have of not being a PhD in biology is my simplified perspective. So many of you should be able to relate to it. For those of you who have a biology degree, I apologize. And if I get anything wrong, please let me know so I can improve it the next time. So can everyone see my slide? Yep. Okay. So this is, like I said before, this lecture is very big picture and very simplified. I will be giving everybody access to the full PowerPoint. I believe that Randy, Jim, and Julie have it, and it's also linked to my Google Drive. So if you want to present this to your church or small group or any other church organization or even a secular organization, feel free to use this in order for us to just show how amazing our DNA is. So when we look at the origin of life, it starts with DNA, and there's two views of thought. One could be it's a combination of inorganic material, add some energy, give enough time and chance, we will develop the blueprint for life. Or is the origin of life come from a elegant designer? If we look at what the Bible says, we go back to Genesis 1, verses 1 and 2, and it says, "In the beginning, God created the heavens and the earth. Now the earth was formless and empty. Darkness was over the surface of the deep, and the Spirit of God was hovering over the waters." Oh, just to let you know, you don't have to take notes because I've given you the PowerPoint. Just sit back and enjoy the presentation. So the question is, what is this word hovering? And if we go back to Brown Drivers Briggs in terms of the Hebrew lexicon, we find that the the word for hovering is rakap, which is only found in one other place, Deuteronomy 32:11, and that's where it talks about an eagle hovering over their young, like brooding. So I like what uh, Eugene Peterson says in the message. He paraphrases Genesis 1:2 as the earth was a soup of nothingness, a bottomless emptiness, and in an inky blackness. God's spirit brooded like a bird above the watery abyss. So what we see here is the Christian perspective is the Spirit of God is brooding over the lifeless waters and is forming the organisms of life. And so the question is DNA and information? What is, well, actually, this presentation is based on these subsets here as an outline. So I'll be discussing whether DNA is an information or a random pattern. We'll look at the basic structure. We'll look at the genetic code redundancy and the machinery within the cell that makes DNA and RNA. Also, we'll look at the self-repairing mechanisms. We'll look at the optimization and compression of DNA. Another thing we'll be looking at is homochirality. I'll explain what that means further in this presentation. And then we'll look at the data storage and archiving properties of DNA. And finally, we'll look at DNA to RNA to hemoglobin. So we'll start off with DNA. Is it an information or is it a pattern? And when we look at information, it is represented in many formats. For instance, music. So if you look here, you'll see the symbolic information of music, and it's a series of notes. And with this symbolic information, we can replicate it and it will be all the same. However, when we implement the symbolic information, we come up with music. So here we have two forms of information. One is the symbolic and then the implementation. Another way we can look at it is information could be done in terms of Microsoft Windows. On the left, we have symbolic information, which is a binary code. But when we implement it, here we have Microsoft Windows. So the question is, information is based on a language, either notes or binary code. We can have exact copies when it comes to information, and it requires elegant thought. So composers, programmers are needed to develop the information. Whereas in a pattern, here we see snowflakes, and they look intricate themselves, quite fine, but we find it comes from chaos. We don't have exact copies, and it requires no thought at all. So next we'll look at the basic structure of DNA. And DNA is made up of looks like a twisted ladder with rungs, and the outside lines are sugar phosphate backbone, and these rungs are made up of nucleotide pairs. I got this slide from Rich Deem, who's also an apologist, and he actually has a really amazing YouTube video which goes into much more detail. So here we have thymine, which matches with adenine, and then guanine matches with cytosine. So these are what we call nucleotide base pairs. Guanine always matches with cytosine, and adenine always matches with thymine. So these are the basis of DNA, and it's quite a amazing structure, which we'll see. So like I said before, they're made up of two pairs, adenine, thymine, cytosine, and guanine. And when we look at simple bacteria, we're looking at least almost 500,000 base pairs. So it's not that simple. It's not just a bunch of codes slapped together. It's actually quite a a large string of codes, nucleotides. And when we look at humans, we're looking at 3 billion base pairs. So if you look at the length of human DNA and you were to uncoil it and straighten it out, how long do you think it is? Now, from a show of hands, how many think it's a nanometer or 10 microns or 2 millimeters or 2 millimeters or 2 meters long? Uh, any show of hands in terms of 1 nanometer? Nobody. Okay, 10 microns. Okay, nobody. 2 millimeters. Okay, the answer is 2 meters long, which is quite amazing because it's such a small piece of DNA that if you were to stretch it, it would be 2 meters long. So if we were to look at all the DNA uncoiled in your body, if we were to put it end to end, how long would it be? If we were to take every piece of DNA in your body, would it be from Sacramento to Paris and back? Or would it be from Sacramento to the moon and back? Or would it be from Sacramento to the sun and back? How many people think that it's from Sacramento to the sun and back? Okay, quite a few people. That's probably a good guess, and this is a trick question because it's none of the above. The real answer is with the 10 trillion cells in your body, it's a round trip from Sacramento to the sun and back 70 times. So just think of that round trip 70 times, and that's the length of all your DNA stretched end to end, which is a distance that's very difficult to fathom. Now, there's a really great website called the DNA Learning Center. And what they've done is they've done a lot of computer CGI graphics, and they've shown what DNA looks like in terms of how it's folded up as well as how it's replicated. So what I'm going to do is I'm going to exit my presentation and we'll go straight to DNA. And first it'll show how the DNA is folded. Because when you think about it, you've got 2 meters of DNA length. How do you get it compressed into one little cell? And after that, we'll be looking at how DNA, this massive 3 billion nucleotide pairs, how do we actually replicate it in terms of how do we regenerate another cell? And this DNA Learning Center shows the process of DNA replication. But first, we'll look at how DNA is packaged. So I'll stop sharing, and then I'll share my other screen.
In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. It is important to realize that chromosomes are not always present. They form only when cells are dividing. At other times, as we can see here at the end of cell division, our DNA becomes less highly organized.
Okay, next we're going to look at
Did you know that
DNA
Amazon has a 50?
we'll look at DNA replication, which is actually quite amazing when you look at the machinery needed to unzip the DNA and then copy both sides of the DNA. One is copied backwards. Just the mechanism alone is almost like a well-engineered machine. We'll take a look at it. It's only a minute long.
Using computer animation based on molecular research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into two strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is two new DNA molecules.
Okay, we'll stop there.
There are robots
Whoops.
the size of beef.
Okay. Let me go back to sharing my other screen. That is just amazing in terms of okay, share screen. Okay, yeah, so to me, that's amazing. When I first saw that in terms of the computer animation, I'm thinking, wow, how does a single cell come up with machinery like that, even for a simple single cell organism? It needs to be replicated, and you've got this helicase and all these other things that spin and replicate DNA at such a high speed. So I find that just totally amazing. Next, we'll look at DNA, the genetic code, redundancy, and also additional machinery that creates the RNA and the amino acids. Whoops. So it happens. I'm not sure for those of you who've been around a while, if you've ever used Windows 3.0 or DOS, what happens when Windows or iOS becomes corrupted? And I remember back in 1990s, you would get something like this, which is the blue screen of death. And that's where your computer hangs up because there's a bit of code that is defective. Or if you're with iOS, we have something called the spinning beach ball of death. Now, I'm not an Apple person, but that's what my friends tell me who use Apple products. You don't want to see this because that means that you've lost your data because something has gone wrong. So what we see here, that DNA is redundant because, as we said before, because the A's and the D's and the C's and the G's, they actually match together only together as pairs. So what that means is one end of the sugar backbone is complementary to the other side. So if you have one side, you can always develop the other side. And once again, here you have the thymine with the adenine, the guanine with the cytosine. So when DNA is transcripted to RNA, they replace the the T with a uracil because the thymine is a lot more costly to make, as well as because we don't need as much energy because RNA doesn't need to be that long-lived in order to create a protein. The body replaces thymine with uracil, and that's actually a good compromise in terms of saving energy. So what we see here is the adenine. So DNA has adenine, so does RNA. They both have cytosine, they both have guanine, but the thymine is replaced with uracil. And this molecule is the deoxyribose. And here you've got a methane molecule, whereas the uracil is just a H molecule. And they both, this one bonds to the double helix, and this one bonds to a strand of RNA. So when we talk about the genetic code, as we talked about before, it's the uracil, cytosine, adenine, and guanine. What the genetic code is, it takes you've got 4 letters, and the genetic code takes any combination of 3. So it could be a UCA or a CG or a CAG or a GGG. And these are what we call codons. And each codon is represented by a amino acid. So because there's 4 letters and 3 letters in a codon, you actually have 4 to the power of 3, which is 64 possible combinations of codons. But because there's redundancy within a genetic code, these 64 codons actually code for 20 amino acids. So let me show you a table here. So for instance, this amino acid called leucine could actually be made up of any of these combinations. So it could be a UUA or a UUG or a CUU. If, for instance, this U is defective and it gets a CUC, then you'll still create leucine. Or if the U becomes an A by accident, it'll still code for leucine. If U becomes a G by accident, it still codes for leucine. So it's quite interesting in the sense that all these, for instance, serine can be coded with these 4 codons, or also these codons here. What's interesting is you have something called the AUG. I can't pronounce it, but it's the start. It's the when it becomes when the DNA gets read into RNA. When it sees a MET, it knows to start creating the RNA. And then when we have the codon UUA or UAG or UGA, it tells the mechanism to stop transcoding the RNA. So it's hard to explain, but once again, I'll go back to the DNA Learning Center, and we'll see how DNA is transcribed into RNA. And then once the RNA is created, some machines come and they actually snip the exons off the RNA to make it translational for the amino acids. So there'll be 3 videos that we'll be watching in sequence. First, how DNA gets translated to or transcribed to RNA, and then right after that, how the RNA is spliced to make it into mRNA, and and then how mRNA is used to create a protein strand. So I'm going to stop sharing and share my other screen. Let's see. Okay, here's the transcription from DNA to RNA. Once again, just notice the mechanisms.
What you are about to see is DNA's most extraordinary secret, how a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the 2 strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.
That I find fascinating. Next, we'll look at how that RNA that was just created is spliced to make it
using the right weight
usable.
method.
Okay.
As DNA is transcribed into RNA, it needs to be edited to remove non-coding regions or introns shown in green. This editing process is called splicing, which involves removing the introns, leaving only the yellow protein coding regions called exons. RNA splicing begins with assembly of helper proteins at the intron-exon borders. These splicing factors act as beacons to guide small nuclear riboproteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the 2 exons together. The edited RNA and intron are released and the spliceosome disassembles. This process is repeated for every intron in the RNA. Numerous spliceosomes, shown here in purple, assemble along the RNA. Each spliceosome removes one intron, releasing the loop before disassembling. In this example, 3 introns are removed from the RNA to leave the complete instructions for a protein.
That just is amazing to me. How does the proteins know how to cut where? I mean, we're talking basic bacteria. These things are splicing the RNA even in basic bacteria. Next, we'll look at how the rMNA is now used to create proteins. This also requires some additional complex machinery that just is amazing. Let's see, let's look at this mRNA translation.
When the RNA copy is complete, it snakes out into the outer part of the cell. Then, in a dazzling display of choreography, all the components of a molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. Each transfer molecule carries a 3-letter code that is matched with the RNA in the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecules. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second. And as a result, our muscles, brain, and all the vital organs in our body receive the oxygen they need.
That is simply amazing. Once again, you look at how the machinery is put together just to translate the RNA into a protein chain. So I will continue with the presentation, and I will share my other screen. Whoops, share. Okay, so when we look at the error minimizing redundancy of our DNA code, some scientists did a random generation of the genetic code for humans, and they came up with 1 million different combinations spit out by a computer. For those of you who are into statistics, you'll recognize that this is a normal curve. And with the normal curve, this is the average. And any of the 500,000 of the codes, the random codes, anything on the left-hand side represents good. And as you go more and more to the left, it becomes even better in terms of the ability to counter substitutional mutations. Whereas on the right side of the 1 million different random codes, these are the worst possible codes in terms of substitutional corrections. So the interesting thing is, of the 1 million random samples of human genetic code, where do you think our human genetic code is on this normal curve? Well, what you find here is our human genetic code is actually right here, which, if you look at statistics, is almost 6 standard deviations away from the mean. So if you remember statistics, if you're 6 standard deviations, that means you're at 99.9999994% of all the codes, where you're actually better than all these codes here. It's just a staggering result of where our human genetic code is. It's not, I would have expected our human genetic code to be somewhere, within 1 or 2 standard deviations, but we're actually 6 standard deviations. And that just is mind-blowing. What that tells us is our human genetic code is highly coded to avoid substitutional mutations. And so of the 3 billion base pairs, unlike Windows, there's no sudden freeze up. Unlike the iOS, there's no spinning beach ball of death. That means if there's a error in one of our nucleotide pairs, the genetic code is enough to make sure that we're still able to function, because as I said before, you've got 64 codons coding for 20 amino acids. And that's just amazing from a coding point of view of our genetic code. Next, we'll look at DNA self-repairing mechanisms, and this is actually even more amazing. They say that in one cell, you've got your 3 billion base pair letters, and every day you'll have about 10,000 lost letters, about 10,000 damaged letters, about 55,000 single strand breaks, and 10 double strand breaks. So that's just in one cell. If you multiply that by 10 to 40 trillion cells, that's a lot of damage just that our body has to repair. And this is where our DNA is just amazing, because it has self-repair mechanisms. So this is a video that I found from the Discovery Institute. It's called The Long Story Short, and I won't show you the whole thing, but I'll show you one part of the self-repairing mechanism. I highly recommend everyone check out the Long Story Short. It is super simple, and it's got some nice cartoons, and it probably has about 10 videos. And I wish I saw this before I put this presentation together, because it pretty well covers everything I want to cover. We'll look at both 2.5 minutes of it, and it's called base excision repair. Just fascinating. So I'll stop sharing, and I'll share my other screen. Okay, let's go there.
But inside of you, there are molecules that can detect errors in your DNA and much more. You can't live without several different repair mechanisms. We've only got time to cover one of the many systems, and the simplest one at that. Here's how it works. The simplest DNA repair mechanism of many is intimidatingly called base excision repair. Fixes a single damaged letter of DNA, and it's needed in every known form of life. If you had a damaged strand of DNA, first, one machine or protein enzyme scans down it and recognizes the single damaged letter. It would detach it from the DNA backbone, extracting it like an old, dirty tooth, leaving the functioning letters alone. Next, another machine detects the empty spot and breaks a single strand of the DNA backbone. A third machine then snips the DNA backbone again, removing the segment of backbone that's empty. These two enzymes have the ability to break the DNA backbone at will. Lucky ducky you, they only do this when needed, and at a specific site of damage. How could enzymes like this just pop up at the start of life without first destroying all of the DNA in sight? A question for the ages. A fourth machine then inserts the replacement part, an intact matching letter of DNA, complete with a new backbone segment. Finally, a fifth machine welds the replacement part to the backbone on both sides, completing the restoration. This is all oversimplified baby talk, of course. The actual mechanics of these things is far beyond what we can convey here. But all this madness is happening every day in every single one of these tiny little danger hot dogs, also known as bacteria. If your reaction to all of this isn't, "What?" then you're just not paying attention. This is literal submicroscopic surgery with the added monkey wrench of the cell does not stop operating while it's being repaired. Imagine trying to repair a Jeep while it's still running. And remember, base excision repair is just one of the many DNA repair pathways, and not even the most complicated one. Even the simplest organisms absolutely need repair mechanisms. The first life and all life needs repair at the very beginning. In bacteria, the instructions to make these 5 enzymes are coded in DNA. requiring more than 8,000 letters of information. If you're smart, you'd notice, just like the scientists at Manfred Eigen, that this is a huge paradox. That much DNA could have never accumulated unless it was first protected by repair mechanisms. But the repair mechanisms themselves require at least this much DNA. This sort of thing is kind of like taking kids hiking in nature. You could probably bring a few of them without losing any. But if you had to bring 8,000 kids on a hike...
Okay, we'll stop there. And yeah, that is amazing when you consider that these proteins are able to cut and repair without cutting other parts of the DNA strand. And also, like you said before, you need this from the very beginning. So how do you get all these repair mechanisms within the single cell bacteria? It's it needs to be there right from the start. So next we will go back to my other screen. That's it for the videos. Yeah, so later on in the video, it says the DNA was designed with the expectation that errors would happen as well as the foresight to be able to be fixed. So within the DNA code, you've got these repair mechanisms that's actually encoded within the DNA. They were saying that 1 in 20 machines in your body are for actually destroying the other 19, and all these components become recycled. I highly recommend that you check out these series of videos. So that, to me, is just amazing that not only is our DNA optimized, but there's actually the DNA provides self-correcting, self-fixing machines within our cell. That. Next, we'll look at DNA optimization and compression. This is also amazing. Some of you have probably seen this before. These are special sentences. A man, a plan, a canal, Panama. Live not on evil. Was that a car or cat I saw? What is so special about these and what they are is we call them palindromes. What that means is if you read it backwards, it reads the same. So for instance, man, plan, canal, Panama, right? But that's not amazing. What's really amazing is what we call heteropalindromes. So these, for instance, reviled, gate man, stressed, when you read it backwards, it becomes deliver, name tag, and desserts. So DESSERTS. That's even more interesting in the sense that as you read it backwards, they form other letters. And what would be even more incredible is if you were to have a sentence read one way, and if you read it backwards, it comes off with a message which even makes sense. So when you think about that, it takes incredible genius to create a heteropalindrome, not just with one word, but a whole series of words, and it makes sense. And that's what we find in certain regions of our DNA. There's actually overlapping codes and what we call different reading frames. So what these codes do is, depending on how you read it, it creates, different binding histone proteins. It controls transcription factors and also machines that splices transcribed genes. So similar to what we saw there in terms of, cutting up the RNA. So how does this work? Well, let's look at one half of the strand of DNA. So here you have the cytosine, thymine, guanine, cytosine, adenine. If you read it 3 letters at a time, it creates leucine and all these other proteins. So that's one way of reading one half of a strand of DNA. But what happens if you were to shift by one letter? Instead of CTG, you now have TGC. Well, guess what? That codes for a different series of amino acids. And guess what? If you were to shift it again by one letter, you now have CGA. Now you have another string of proteins. So that's only half the strand of the DNA. You also have other half, which has the complementary letters. So by reading the strand in one direction, you can actually have up to 3 different protein strings. If you read it the other way, you have another protein string. You shift it by another letter, you have another one, different proteins, and then shift it one more time. So you have 6 reading frames. You can have actually up to 6 different protein strings. And here, as we said before, you have the MET. So when the machinery sees the MET, it knows to start coding for the RNA. And also here we have the stop codon. So as you're shifting the reading frames, you're getting different codons combinations. And that is, I'm not saying that the whole DNA is like that, but there are certain sections within our DNA that allows up to 6 different reading frames. And that's what I'm talking about, heteropalindromes. You know, you've got DNA being read in one direction, but then you could read it in a different direction and actually makes sense. It's not garbage. It actually creates a usable protein and machinery. That, in my mind, is totally amazing. One of the things that Francis Crick, when he looked at the DNA, he didn't quite understand it, and he said, well, I can see, 3% of the DNA coding for proteins, but the other 97% seems to have no function at all, and it must be leftover baggage from evolution. And as a result of that, no one has really looked at this so-called junk DNA, and that's really hindered our genomic research. Now, back in 2012, there's the ENCODE project, and what they've done is they've analyzed our DNA, and they've said that they think that 80% of our human genome is actually transcribed and is functional. And what that does is it handles the switching signals, gene regulation, and it provides some coding for fighting viral infections. Now, some critics, actually a lot of the critics question whether it's 80%. The reason being is if it's 80% functional or even higher, that means their theory of evolution, having all this baggage coming from the first the single cell, as it evolves to humans, we're carrying baggage. But if we find out that a lot of the DNA is functional, that means it's not carried on from our previous ancestors, but it's actually programmed to be functional. And so what I see here is our DNA is not junk, and we would predict that if it is coded with such incredible design, we would say that most of our DNA, not just 80%, but close to 90, 95, or 99, almost 100% of our DNA is functional. Next, we'll look at homochirality. And homochirality means, homo means single, and chirality is handedness. So when we look at molecules, even though they have the same chemical formula, the sugars, you could have a right-handed orientation or a left-handed orientation. The same way when you look at your hands, you've got your right hand and left hand, you got one thumb each and 4 fingers. They look the same, but they're a mirror image of each other, and they can either be left-handed or right-handed. What we see here is on the DNA backbone, these are made up of right-handed sugars. So the molecules that make up the DNA backbone are all right-handed, which is incredible. Also, the left-handed amino acids that build a protein, or all the amino acids, they're all left-handed, which is a problem because in real life, in the probiotic soup, you have both equal left-handed molecules as well as right-handed molecules. The question is, how does nature know how to isolate all the right-handed molecules to create the sugar backbone, as well as all the left-handed amino acids to build protein? And it's just very difficult to do. So what I've done is I've done a little bit of audience participation. If you have your birthday on an odd date, please put up your hand. Okay. So if you're on an even date, leave your hand down. So if you look at everyone around you, see if you've got a contiguous left hand in terms of how many left hand people do you have in a row. And if you have, 100 people, you'll find that it's very difficult to get, 100 people all in a row to be left-handed, let alone at most maybe 3 or 4 to be contiguous. So when you consider having all left-handed molecules or all right-handed molecules forming a single chain, that's just near impossible because, for simple bacteria, you need a right-handed chain of almost 500,000 uh, uh, 500,000 sugar molecules for the backbone. And then for humans, you need almost 3 billion uh, right-hand sugar molecules for the DNA backbone. So when you look at it from a time and chance possibility, it becomes less and less possible the more the more base pairs you have within DNA. So from a homochirality point of view, the fact that our body is able to put all these right-handed or left-handed amino acids together, that's just amazing. Next, we'll look at DNA storage. How are we doing for time? Well, we'll get it done. DNA storage. So when you look at all the data in the world, if you include YouTube, Facebook, Google, Netflix, the entire World Wide Web, all the genomic research, WhatsApp, Instagram, that's a lot of data stored in servers, on floppy disks, everywhere. That's equivalent to more than 900 DVDs per person. That's the total data storage in the entire world. So what's amazing is the total data capacity of the 10 to 40 trillion cells of DNA in your body is equivalent to the entire world's database, data capacity. So for instance, in 2020, we had 40 to 60 zettabytes, that's 21 zeros of data stored. And within the human body, within the DNA, the 10 to 40 trillion cells that you have, or DNA strands that you have, that's equivalent to 60 zettabytes of information. So if all your DNA could be used to store data, you would have the entire world's database stored within your body. That's totally amazing in terms of the data storage capacity of DNA. Also, when we look at data archiving, some of you probably remember magnetic tape back then, and then CDs, probably now out of not used, probably still using DVDs. We're also using a lot more flash storage, SSDs. Unfortunately, they don't last that long, especially if you have DVDs or flash storage. You're looking anywhere from 10 to 25 years. So if you've got your wedding photos or photos that you want to save, probably be best to make sure they're backed up to the web or something, because they don't last forever. They tend to degrade over time. What is amazing about DNA is if you were to take DNA and encase it in glass, stored at normal temperature, it would last for about 2,000 years. If you were to store it in a cooler temperature, it could last for up to 2 million years. Right now, scientists are actually using DNA to code data. And what that tells us is the data archiving and the data storage capacity of DNA is just amazing. Finally, we'll look at RNA production. So we touched about this before in the video on how the RNA was translated into hemoglobin. So when we look at RNA, how many blood cells are produced in our body every second? Is it 24,000, 240,000, 2.4 million? Well, the answer is 2.4 million. So every second, your body is creating 2.4 million red blood cells, right? But that's not amazing, because what's really amazing is within a hemoglobin or within a red blood cells, how many hemoglobin molecules are there in a red blood cell? Okay, so previously we had, 2.4 million red blood cells per second. And then in each red blood cells, how many hemoglobin molecules are there in one cell? Is it 270,000, 2.7 million, or 270 million? Well, it really amazes me that you need 270 million hemoglobin molecules within one blood cell. That's just one blood cell. And so what that means is of the 270 million hemoglobin molecules per one blood cell, 2.4 million blood cells per second, you're actually producing 100 to 600 trillion hemoglobin molecules every second. So just think about that. Your body in 1 second just created 600 trillion hemoglobin molecules just based on that DNA being translated into hemoglobin molecules. But that's not amazing, because what's even more amazing is this is the molecular formula for hemoglobin. It's almost 3,000 carbon atoms, about 4,500 hydrogens, about 800 oxygen, 800 nitrogens, 8 sulfur, and 4 iron atoms. So what your body has to do is it actually has to put together all these atoms just for one hemoglobin. So what that means is you've got 2.4 blood cells per second. Whoops. 270 hemoglobin molecules, and for each hemoglobin molecule, you've got almost 9,000 atoms per hemoglobin, which means that your body is arranging 1,000 to 9,000 quadrillion atoms per second. I mean, when you're tired, that's probably the reason why you feel tired, is your body is manufacturing, arranging atoms every second. This is not just in a year or for your lifetime. This is every second your body is arranging these atoms. And that is amazing. So in conclusion, when we look at the DNA structure, we see that the genetic code is redundant. It's got self-repairing mechanisms, highly optimized, highly compressed with up to 6 different reading frames. It's all homochiral. It's all left-handed, right-handed sugars for the backbone, left-handed proteins for protein chains. The storage capacity, your body is equivalent to the entire world's database. The archiving stability can last anywhere from a couple thousand to a million years. The RNA cell factory, or the hemoglobin cell factory that the RNA and your whole body is producing, is just amazing. So when we look at DNA, we see that it definitely is information, because it's based on a code, and we can have a lot of simultaneous codes identical, multiple copies, which points to an extreme elegant programmer and construction. It kind of reminds me of what we see in Psalm 197, and it says, I praise you because I'm fearfully and wonderfully made. your works are wonderful. I know that full well. So as a Christian, when I want to praise God, I think of how He's put our body together, how He's put life on earth, and it is just fearful and awesome in terms of how intelligent and how our body is made. So, when I hear people saying that, yeah, we evolved from from material and some energy, I look back at how amazing our DNA is, and it snaps me back to uh, that the Christian worldview explains the origin of life much better than any other worldview. Period. What I'd like to also encourage all of you to do is to watch the other long story short videos. These are really fun to watch. We saw a bit of it, and they're actually very funny, but it's a lot of good information. Like I said, it's put out by the Discovery Institute, and yeah, just do a search on YouTube under long story short. Also, I highly recommend that you check out the DNA Learning Center. I've shown you probably about 5 of many DNA CGI videos, and some of them are simple, some of them are really complex, and it gives you a real appreciation of what our DNA does. And also, I'd like to give credit to Richard Deem, who he's given me permission to use some of the slides. He has a very in-depth thing on the designs of DNA, which is even more complicated, as well as more correct in terms of his presentation for DNA. So like I said, I've given the full PowerPoint to Randy, Jim, or Julie. If you want to use this for your own purposes, go right on ahead. We want to use it to bring attention to our elegant designer. Just make sure you retain the credits to Richard Deem. So I will stop our presentation there, and I'll take some questions. Now, like I said, I'm not a PhD in biology, but I'll do my best to answer some of your questions. In fact, some of the other apologists should be able to help me out as well.
You want to come up and ask questions, please do. Yeah, come on up.
Here we go, somebody coming up. Hi, my name is Brian. I'm just wondering, everybody wants to go to Mars these days, and we no longer have radiation protection. So what's that going to do to our DNA over years of experience, and then our children? I mean, if somebody's going to try to have kids on Mars, how mutated will the children be?
Yeah, I spoke to Hugh about that, and Hugh says that don't sign up to go to Mars. You'll be dead by the time you get there. Your digestive system will be totally destroyed unless they find some way to shield your body from the deadly cosmic rays. Yeah, I mean, right now, our Earth is being protected by this magnetic field, the dynamo, our spinning liquid metal core that's protecting us from the cosmic winds from the sun, as well as the cosmic rays. But once you're once you're on a rocket ship to Mars, you no longer have our magnetic field that's protecting you. So unless you're carrying lead with you, which is not a good idea to have lead shielding in a spaceship, because we want to have spaceships as light as possible. Hugh says that, yeah, you're going to die by the time, well, you'll be severely in bad shape. Just your digestive systems will be totally mutated, and yeah, you won't be able to survive on the planet of Mars unless they find some way of protecting you. So
Yeah. Okay.
Thank you for an excellent presentation. Can you comment? I know the naturalist will say this all occurred by chance and time. But from what I understand, the specific base pairs considered what's out in nature are highly selective for utility for DNA. Do you have any comment on that? The purines and pyrimidines are
I won't be-
exact right ones that are needed?
Yeah, I can't answer that one. That's what was the question again, Randy?
Well, the guanine, cytosine, adenine, and thymine are the exact right base pairs, considering the hundreds of possibilities out in nature. That they are the ones that actually work for DNA, where other ones would not work. So if we were speculating on some sort of a random process where these base pairs came together to form some type of a coding system, we've got the right one, which I think argues again for design rather than a randomness.
Actually, that's a great point. I wasn't aware of that. And it just once again points to the incredible designer putting it together. So what's the chance of, getting a base pair to hook up, let alone, up to 500,000 nucleotide base pairs? I mean, as we've seen before, you not only need the right base pair molecule, but you need error correcting, you need a backbone that is right-handed. All these, when you look at the weight of evidence, just points to a materialistic evolution being, or in my opinion, less and less probable for that to happen.
One other follow-up question, Lester. This seems very compelling as you're presenting it. I can't imagine this happening by chance. What has been your experience when you've presented this to skeptics? Have they taken a second look, or do they try to just dismiss it as not important?
I spoke to one person who was a biology major, and he was trying to explain to me, no, there's other processes that can account for that. What is interesting is I just finished watching a video, and they were saying that all this junk DNA, a lot of the critics, just recently they're finding that the endoretroviruses are sections of the junk DNA which they thought were junk are actually functional. So, I mean, some of the stuff that I showed you, I left out. For instance, even the metabolism. If you look at the decision tree for metabolism, that is just super complex. Yeah. Well, I guess when you talk to atheists, they don't want to admit that there is a designer, not because they see the argument, but by having a God who exists and someone they're accountable for, that is enough to look for any other excuse to deny a causal agent. So I can understand from a philosophical view why they don't want to accept it, and they will grasp at any excuse to deny intelligent, design, an intelligent designer. But I mean, they're as I finished watching, if the DNA is 80 to 90% functional, that pretty well destroys evolution in a sense that evolution is not a evolution of baggage carried from the single cell organism. If our DNA is finely tuned and used for every aspect of our life, that means that, yeah, that evolution becomes less and less possible. So getting back to your question, most people I show this to are amazed. They're not even aware of the homochirality. They're not aware of the reading frames. They're not aware of the self-fix mechanisms. But some of the people that I did talk to will cling to anything just to avoid the conclusion that we're accountable to some causal agent that knows more than we do, and that we're accountable to. So I think the argument or the conflict is more philosophical than science based.
Come on up.
About cross-species fixing.
Why don't you come up, Brian? And anyone who is on Zoom too, we'd love to have you ask questions as well.
I was just wondering about cross-species DNA. How, you know, they're so specific to the species, you would think they would never mix.
Yep. Well, that's the, yeah, actually, I just finished watching a video about that. Again, we actually share common DNA coding, you know, with daffodils. Actually, we share more with daffodils than we share with other mammals. So does that mean that we, you know, evolved from daffodils? What that shows is it's not excess baggage that we've carried. It actually points to a common designer.
Right.
Same
Yeah.
way that an engineer can design multiple cars, you know, different models with the same wheel, same steering wheel, but different body designs.
Right. Yeah.
Yeah.
Thank you.
Mhm.
Hi.
Yeah.
You mentioned, well, in short order, you convinced me very thoroughly that I should not raise my hand to, under current technology, to take a trip to Mars. I wonder if it's that dangerous out there. What's happening to our astronauts that have accidentally been out there for an extra 6 months?
Well, actually, if you look at our ISS, International Space Station, we're actually still shielded by our magnetic field. So, yeah, if you look at the globe, if you take your normal globe, the orbit of our space station is only less than an inch above the surface of a normal home globe. So from that point of view, people who have been in space for 6 months or longer are still protected by the Earth's magnetic field. When the astronauts went to the moon, I think they were only there for less than 4 or 5 days, less than a week, and they were concerned because they left the Earth's magnetic field. And so if you were to have a base on the moon, what you'd need to do is you need to go underground, find some cave to protect yourself. And probably the same thing with Mars. You'll have to, you won't be able to build a structure on the surface. You'll have to go subsurface. But the problem is getting there. It's almost yeah, months or even a year, depending on where Mars is in relation to Earth's orbit. Like Hugh said, you will have severe mutations within your body. Yeah, it'll destroy your digestive system. So it sounds great to be able to move to Mars, but probably not the best idea at this time.
So what you're saying is we have to invent Star Trek's transporter first.
Yeah.
When they do that, can they fix my cavities on the route?
Yeah. Transporting probably just as hard as warp drive. Warp drive is impossible. Just to warp space, you would need to convert the energy or the mass, or if you go E equals mc squared, you'll need the entire mass of Jupiter every second converted to energy in order to warp space. So, yeah, the stuff that you see on Star Trek or Star Wars, hyperdrive, warp drive, it's impossible. Too much energy involved. And I actually, Rich Deem also does another interesting thing. If you're traveling near the speed of light, we're not talking warp speed, but sub-warp speed, as you're going through space, if you remember the Doppler effect, when an ambulance is coming towards you, the sound waves get compressed, so you hear this. Well, the same thing happens to light as you're traveling through space. As light is coming towards you, as you're speeding towards sources of light such as other stars, you're compressing the light waves to become near X-ray and cosmic rays, so they become deadly. As well as if you're traveling through space at near the speed of light, if you hit a speck of sand or a little dust particle, that's equivalent to having an impact of an atomic bomb against your shielding. So yeah, for one thing, you can't hit warp speed, and even if you're near light speed, the compression of the light waves will kill you, as well as any dust particle or any sand particle will totally destroy your spacecraft. So the likelihood of getting to Mars in a timely manner is near zero.
So I guess the bottom line is we're never leaving this planet.
Yes, yeah, and
Yeah.
yeah. Now, this planet is probably, as you probably know, of all the given all the stars and galaxies, the trillions and trillions and trillions of planets and stars, this planet here is probably the only planet that will sustain advanced life in the entire universe. So that says something in terms of how God has created this beautiful planet that we live in uh, to provide a place for us to live.
What kind of engineer are you?
I'm a civil engineer. I design airports and runways and port facilities.
Oh, okay. Yeah, I invent machines.
Oh, okay. Oh, I also design highways too. Yeah.
Good. Yeah, make them out of old tires. Get rid of all the old tires.
Mhm. Yeah.
That's cool.
Here comes somebody else.
Yeah, my name is Cliff Bracken, and kind of a little bit off the track here, but
Mhm.
about a year ago, I had a stroke, and I've done a lot of research into health. And what I have found is that we are, in my opinion, elegantly, efficiently designed to die. And everything in our science says, about 120 years is max.
Mhm.
Yet, we have in our body these stem cells. And from everything I've read about the stem cells, they have no purpose except whatever we need, they will become that part of our body,
Mhm.
which some scientists think we have the potential of living many years. So I'm just kind of curious, this paradox. Do you have any opinion about that, or what is your thought?
Yeah, my thought is, yeah, would it be nice to live for over 120 years, maybe 200 years? But when you think about it, eternity is a long time. So, even if we were to double our lifespan to 240, we're all going to die eventually. No one's going to be living to 100 or even a million years. But the thing is, for certain, we're all going to meet our maker at some point. So, yeah, whether sooner or later, in my opinion, even if we double our lifespan, we still have to face our maker. So, yeah, I mean, like Hugh says, the next realm would be far better than you can ever imagine. I mean, no eye can hear, no eye can see, no ear can fathom, or nobody can fathom what the next realm will be. So the thing is, I mean, our Earth will be past the Goldilocks zone, you know, all those zones for advanced life, probably within 100,000 years, will be, you know, past the sun's window of habitability. So guaranteed, even if we live up to 100,000 or 200,000, our Earth will not be habitable for advanced life. Or so, yeah, I could be wrong. It's either half a couple 100,000, but for sure, our Earth will not be habitable in a million years. So it all depends on your time scale, you know, whether we're talking hundreds or thousands or millions. The nice thing is eternity is a long, long time. And that's the nice thing about the Christian worldview is our destiny is secure, regardless of whether we are able to extend our life here on Earth on a temporary measure.
Well, what's interesting though, and I guess that's what I was trying to point out,
Yeah.
the stem cells are not limited as to what they can do. They're, you know, it was like God put into us this ability to transform any organ or create things. And I don't know, it's just interesting that these stem cells are so uniquely designed. They have no purpose but can serve any purpose, at least, you know.
I'm also reminded about what Hugh or Fuzz said in terms of our telomeres. They're kind of like the caplets of at the ends of our DNA. Even though you clone sheep, it doesn't. The telomeres become shortened, and that's something that you can't fix. So, yeah, I believe I could be wrong, but even if you were to use stem cells, there's something about our telomeres that limit our lifespan. Now, not only do you have to, you know, contend with the stem cells, but you know, we still have cosmic rays, we still have radiation, you know, from igneous rocks that are hammering our body, creating a lot of damage. So, yes, we might be able to extend our life, but not by a significant amount in terms of eternity. Yeah, but that's a great question.
I recall when they cloned Dolly, the little sheep, they had like a baby sheep that they cloned. Well, great. But as it turned out, they found out the sheep was actually the age of an old sheep. So the telomeres were a difficult thing to get past. Just because you have a new sheep doesn't mean it's not about to die of old age.
Yeah, that's what I heard too.
The other thing I remember something about the anti-anthropic principle, which is, you know, how the answer to the question, how long until the Earth is no longer habitable? I believe some years back, RTB was placing that at 40,000 years, as well as some other non-Christian scientists that were not Christians. So there's a little FYI there.
Okay, so 100,000 probably in the right ballpark within magnitude. Yeah, from what I understand, there's about 10 Goldilocks windows that need to be perfectly aligned. So right now we're all perfectly aligned, but as time goes on, the windows will, you know, will no longer overlap.
Jim, was there something in the chat? In the chat?
Okay, somebody had said something, yeah, all right. Hey, you want to read that? Second Thessalonians 2:9 speaks to why non-believers reject the obvious truth of creation. I have to look it up, but I can imagine what that's saying.
And please, anyone who is on Zoom, please unmute yourself and ask a question. We would love to hear from you. Or Bill Payne with that reference, if you'd like to unmute and expand further?
Mhm.
It just says that for those who refuse to believe the truth and so be saved, God will give them a strong delusion so that they may believe what is false.
Hmm.
Okay. Very appropriate. Thank you.
Mhm. Thank you, Bill.
Other questions for Lester? This is a great opportunity. We have him here, so please. Brian's coming back. Okay.
Okay.
Watch out. I'm curious about, when Adam and Eve and the early people were living towards 1,000 years. And then
Mhm.
suddenly they were only living to 120 years.
Yeah.
Was that God changed the DNA, or was that a change in like the magnetic sphere around the earth and suddenly we have more radiation coming down? Or you have any idea why the sudden change other than, I don't know, God dictated it?
Actually, there's two reasons for that. Hugh talks about. One is astronomers have discovered a supernova that exploded probably about 40,000 years ago, and that was the time that caused damage to the DNA, and it shortened the lifespan, as well as when Adam and Eve during the time of Noah, when they moved away from the Mesopotamian plain, which is predominantly sedimentary rocks, and moved throughout the world, they're now exposed to igneous rock, which has a lot more trace elements of thorium and uranium, and I think potassium. But as we get away from sedimentary into igneous rocks environments, we're now exposed more to radiation. So in combination with the deadly cosmic rays from the supernova, which covered and bathed the entire Earth, as well as our environment, that is probably one reason why the longevity, has been reduced from, hundreds of years down to 120.
Okay. I just recently found out that Saudi Arabia is quite a bit volcanic.
Hmm.
The landscape and everything. They just had the Dakar rally across there, and I mean it's amazingly volcanic. So you kind of wonder if what you're saying is true, that all this exposed rock is, part of the problem.
They're talking about having an experiment where they would take a deep salt mine and then, send people down there to live forever to
Oh
see
boy.
if that would extend their lifespan. But so far, no one wants to volunteer to live deep in a salt mine to avoid the radiation and the deadly cosmic rays.
Yeah. Thank you.
Okay, you're welcome.
Anything else?
Yeah. Yeah, I've got a question. One thing I get a little confused about is the DNA itself. Maybe need a bigger picture here of what's going on. I've always wondered, so do you just have one, in a particular cell, do you just have one huge, long, 2-meter long strand of DNA? Or is it like in different pieces, or how does that work?
No, you get one big long strand of DNA. So
And then okay, yeah. And then when they show the chromosomes and stuff like that, is because they never show them connected. So I always wondered how that worked.
actually, that's a good question. Yeah, because sometimes when you see it, they look like they're all in pieces, right?
Yeah, yeah. How does
I wonder, I think maybe they are disconnected, but it is one long strand, maybe cut up into separate chromosomes. But from what I understand, when you have cell division, you're creating an exact copy of all the single strand of DNA. Now, I could be wrong. I'm not a biologist, but that would probably make sense to me, that if you were to take all the different chromosomes and line them all up, yeah, you would get your 2-meter strand. But yeah, it kind of makes sense that it would be discrete pieces, because otherwise, man, try unwinding, you know, a 2 meter long strand.
Yeah, I was just wondering about that. And also there's the mitochondria DNA. Do you know how that fits into the story here?
And no, that's something that you'd have to ask a biologist.
Okay. All right, that was fine.
Great,
Good.
thanks.
Yep, thanks.
But that's a good question. I'll have to investigate that.
All right, well, thank you. Any other questions? We have time for a last question. Is someone on Zoom or someone here wants to ask something? Oh, Julie's coming up. Okay. The last call, we got her to come up.
Just a question about the vaccines that they say that they're the mRNA, you know, how they vaccines, do they affect the DNA? Like that's a question that was going around, or do you know?
That's an easy answer for me. I have no idea.
Okay, okay.
But like I said, I just want to encourage you to take that PowerPoint. It's free. You could use it however you want. Change it. Just use it in order to, promote the awesomeness of our creator.
Well, Lester, thank you again for your time and expertise. It's been a wonderful presentation. I think we've all been amazed, to use your words, at what God has done in DNA, and hopefully it will be a talking point forward for us as we share who he is and what he's done, and just the magnificent creation that he's made for our bodies. And we just even the hemoglobin was one small molecule. You multiply that by thousands of molecules and how things work together in ecosystems within our body. It's truly remarkable. So again, thank you so much for your time, your expertise, and sharing with us this evening. And we hope you warm up in Canada.
Actually, it's great. Yeah, it's not this isn't cold at all.
Send it all to Atlanta.
You used that. So those of us who are in the room will have a little bit of time to spend together. Those of you who were on Zoom online, thank you so much for joining us. We hope you will come back again. Next month, we'll have Mark Perez here. Mark is an apologist with Reasons to Believe, and he co-authored a book with Ken Samples called Clear Thinking in a Messy World. And he will be speaking next month on cognitive biases. And we all have them, and how we can recognize them and hopefully avoid them. So I think it's going to be a very interesting talk as well. So please mark your calendars it's always the third Tuesday of the month in the evening at 7 o'clock here, Pacific time. So we'd love to have you all back. So thank you, and have a good evening. And again, thank you, Lester.
Okay. My pleasure. Okay. Okay, have a great evening. So long.
Thank you. Thank you.
Bye-bye.